The small intestine is the workhorse of the system. The entrance to the small intestine is controlled by the pyloric sphincter, which opens and allows small spurts of the contents in the stomach to enter the small intestine. The exit from the small intestine is at the ileocecal valve. So this isn't controlled by muscle, but it does prevent backflow from the large intestine into the small intestine. And over here, you can see the ducts from the liver and from the pancreas and how they empty into the upper part. The first part of the small intestine is the duodenum. Duod means 12. And so this is 12, they called it fingers back then, but it's roughly 12 inches long. And despite the fact that it's so short compared to everything else, there is more digestion and absorption that occurs in the duodenum than any other part of the GI tract. The next part, I forgot to say that the duct from the pancreas and liver and gallbladder empty there. But the next part is called the jejunum, and after that is the ileum, which is how we get the ileo in the ileocecal valve. Now, the function of the small intestine overall is the same as we move from duodenum to jejunum to ileum, but there are some changes because we expect the molecules we're digesting to be larger up here, but as digestion continues, they're going to be smaller. So the function of the intestine sort of transitions to match what the process is inside. All along the small intestine, the surface is covered with villi, which means little fingers, and these help to increase the surface area. So this is a villus, a cartoon version. This is a villus, a picture taken from a light microscope. And this is a picture of villus looking from the above, looking at the surface. And you can see why it's called little fingers. Kind of looks like how you might imagine a carpet if you were to magnify it. So this increases the surface area enormously. Now, the villi are about 1 millimeter long, so don't get the impression that that actually looks like little human fingers. To the naked eye, 1 millimeter is not that much. It's going to look like a kind of fine fuzz. The villi contain blood vessels, you can see those, and lymphatic vessels, that's what the green is here, for carrying away the absorbed nutrients. So as stuff gets absorbed into these cells, it can immediately hop into the system that's going to carry it away. The surface area is also super important for carrying out the final step of digestion, not just absorption. Because as we'll see, along the surface of these cells are also enzymes that make sure that we complete that last step. Now, even the villi have villi. This is how important surface area is. So if we take that villus that we were just looking at and we magnify it, so this villus is now this villus. And then we magnify a section of that again. Now we can see that these cells on the outside of the villus themselves have little fingers called microvilli. And the microvilli increase the surface area even more than the individual villi do. The microvilli have tons of membrane proteins. So if we take a closer look at the villi, here's of course the cartoon version. Here's an electron micrograph as opposed to a light microscope. The proteins that we see on the outside of the villi, there are two main kinds. One is enzymes. Enzymes complete the last step of digestion. So when a molecule that has been mostly digested, maybe we're down to a dimer, it encounters the microvilli, it will land on an enzyme which splits it into the two monomers. And then the transporters, which are also all over the place, bring in the two digested monomers. Then we've completed, well, not completed, but done the absorption process as well as the final step of digestion. Now, all of this takes teamwork. Being able to absorb substances effectively, it requires a lot of specialization. Being able to secrete a lot of substances requires a degree of specialization. We need different kinds of cells. So in order to make this all happen with maximum efficiency, the small intestine does absorption, the liver and pancreas do secretion. The small intestine does some secretion too, but by and large, it outsources those functions to these other organs so that it can do a better job of its main task. So it's because it'd be difficult for any tissue to do both of those things well. The pancreas is going to secrete enzymes and bicarbonate, and the liver secretes bile and micelles. Let's take a look at the liver, because understanding what goes on in the small intestine requires understanding what's being delivered to it. The liver secretes something known as bile acids. Bile acids are important for emulsification of lipids. This is a form of mechanical digestion that I'll talk about in just a moment. Bile acids are actually pretty expensive to make. And so they are secreted by the liver, but then after they do their job, they're reabsorbed as much as possible from the small intestine and transported back to the gallbladder so they can be used again or recycled. Micelles are like little vesicles that are important for transporting fats out of the intestine. Fats we have some difficulty with because fats are not soluble in water, which is what we're made of. So the structures for moving fats through the system are different from the other molecules. Also, the liver has a really important role post-absorption. So after you've had your meal and after it's been absorbed, all of those nutrients move to the liver, and the liver processes them. It makes sure that anything that's toxic is detoxified. If the body doesn't need more of a particular nutrient, then the liver will store it. It will pass things on that the body has too much of. So it's sorting and storing after we absorb stuff, because we don't always have a use for what we're consuming. The pancreas, which also empties its contents into the duodenum, secretes about 1.5 liters. Oh, the liver does about 0.5 liters. So 1.5 liters of all kinds of crazy stuff comes from the pancreas. It secretes a number of inactive enzymes. It secretes pancreatic amylase, pancreatic lipase. Remember, the amylase is for digesting carbohydrates. Lipase is for digesting lipids. Ribonuclease and deoxyribonuclease are for digesting RNA and DNA, respectively. And then we have trypsinogen, chymotrypsinogen, and procarboxypeptidase. All of these, as they are produced in the pancreas, are inactive. But the ones that are blue become active once the pH is neutral. And the ones in green become active after reaching the duodenum, where there's an enzyme that turns them on. So first, let's talk about the pH part. So one of the other things that the pancreas secretes is bicarbonate, sodium bicarbonate. And bicarbonate, if you'll recall, is a base. And the stomach, if you'll recall, is producing acid, which is fine for the stomach, but the rest of the digestive system is not too keen on it. So the fact that the pancreas is producing bicarbonate means that when the pancreatic juice mixes with the stomach acid in that first part of the small intestine, the pH will become neutral. The acid and the base cancel each other out. And as soon as the pH is neutral, these enzymes refold properly and become active. Now, also in the duodenum, the small intestinal cells produce an enzyme called enterokinase, which you see here. And so as soon as these green enzymes arrive in the duodenum, they encounter the enterokinase. Enterokinase removes a little piece of the trypsinogen, and it becomes trypsin. Trypsin then activates chymotrypsinogen into chymotrypsin and procarboxypeptidase into just plain old carboxypeptidase. These guys start digesting proteins. So basically, at the very beginning of the small intestine, we are finally digesting all 4 categories of the organic molecules. In addition to all that important stuff, the pancreas also acts as an endocrine organ where it secretes hormones that help to regulate blood glucose. The most famous of these is insulin, but it also secretes one known as glucagon and another one that helps to, all 3 of them together, help to keep the blood glucose levels stable and not fluctuate too wildly. Okay, so we've got all this stuff coming into the duodenum, and now we need to talk about how mechanical digestion in the small intestine occurs. Now, there are 2 types of mechanical digestion in the small intestine. The first is emulsification. This is the process of splitting a larger fat globule, I love that word, into smaller fat droplets in order to increase the surface area for chemical digestion. So you've probably seen emulsification, whether you realize it or not, when you look at salad dressings, for instance, at the grocery store. A simple salad dressing is oil and vinegar. And if you just put those 2 substances together, they separate out into 2 distinct layers. But salad dressing manufacturers have added emulsifiers, which break the fat down into little droplets so that it will stay suspended in the dressing and not separate out. So the other kind of mechanical digestion is called segmentation. It's a method of mixing enzymes with their substrates by contracting the circular muscle of the intestine. So imagine that you have this long tube of stuff that needs to be mixed and you were just squeezing it at different places along the outside. That is exactly what the small intestine is doing. All right, let's take a look at emulsification. Emulsification, at first appearance, looks like it's chemical digestion, but it's really not. So here we have a lipid globule, a big drop or clump of lipids, and the enzyme that we have for breaking down lipids, called the lipase, tries to digest it. Now, that is a big piece of fat to be digested, so it's going to take the enzymes a long time to sort of chew their way into the interior because there's just not enough surface area accessible. So, to the rescue comes the bile acid. And bile acids are able to do their job because they have a hydrophilic side, water-loving, and a hydrophobic side, or water-fearing, which means that when they interact with the lipid globule, the fat globule, they are able to break it into smaller and smaller lipid droplets. Now, the lipase enzymes have much more surface area and they can complete the digestion way more efficiently. Peristalsis is not the same thing as segmentation. Peristalsis uses both the circular and the longitudinal muscle for forward propulsion. In this video, you can see a substance being pushed from one end of the small intestine towards the other end of the small intestine. Now, pancreatic enzymes do the majority of digestion. The pancreatic amylase digests carbohydrates. Proteins are digested by trypsin, chymotrypsin, and carboxypeptidase. Nucleic acids are digested by deoxyribonucleosidase and ribonucleosidase. And lipids are digested by pancreatic lipase. Now, most of this is happening in the lumen as it's being mixed around. But the job for all of these is finished by membrane transporters on the apical side of the intestinal cells. So recall, whoops. Pancreatic enzymes do most of the work. In the lumen of the small intestine, carbohydrates are digested by pancreatic amylase. Proteins are digested by trypsin, chymotrypsin, carboxypeptidase. Nucleic acids are digested by the various nucleases. And lipids are digested by pancreatic lipase. However, the final step in digestion is typically done by enzymes that are located on the microvilli of the intestinal cells. So as the substances, as trypsin, for instance, breaks down proteins into smaller and smaller oligopeptides, eventually we get to a dipeptide or just 2 amino acids, and this enzyme can finish the job, which allows transporters to bring the amino acids into the cell. So the small intestine absorbs more than just those monomers, the amino acids, the nucleotides, the fatty acids, and who am I forgetting? Monosaccharides. The small intestine also is where we absorb vitamin B12 that is connected to intrinsic factor. If those substances, if the intrinsic factor doesn't bind vitamin B12, then B12 cannot get across the intestinal lining. And B12 is very important for red blood cell synthesis. You can end up with a very severe case of anemia without enough B12. So